Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. Well, Jeremy, it's super cool to have this chat with you as we build up for the 2026 Cancer Neuroscience Symposium. We like to start by asking a little bit about you, the person, your background, and I guess I understand that you're at the Cold Spring Harbor Labs in New York, and so how did you arrive in New York, and do you consider yourself a New Yorker now, or what are your thoughts about living there and working there?
So I'm an assistant professor at Cold Spring Harbor Laboratory on Long Island in New York. I have a sort of roundabout way that I've sort of come about working at Cold Spring Harbor. I got my undergraduate degree in anthropology, which is totally unrelated to cancer, obviously, or neuroscience. And part of that involved me going and working with wild animals, and I got very interested in animal behavior. But I realized that I couldn't really study animal behavior unless I could tinker with the tissues and the organs that control behavior, and that's the brain and the nervous system. And so I essentially used that rationale to convince a graduate program to let me in for neuroscience. And then when I was a graduate student in neuroscience, I was in a big building called the Biomedical Research Tower. So there were like 50 labs in this building. And so I would frequently interact with people that were working on things that I didn't even know about, including cancer. And I attended a couple of talks from nurses and physicians that were treating patients with various kinds of cancers. And the common theme that came up across malignancies was really the subjective experiences of having cancer and the problems that people have that are sort of secondary and that doctors don't really think about too much, like fatigue, things like changes in appetite, changes in inflammation in your body. There's changes in cognitive function that are quite common across cancers, and they seem to get exacerbated with cancer treatment. Also, things like pain. And so that got me very interested in how could the nervous system, which controls all of these things, be regulated by cancer, and reciprocally, how can cancer regulate the nervous system? And I just became sort of fascinated by that question, and that led to my dissertation research on how breast cancer influences the circuits in your brain that control sleep and arousal. And that sort of happened to be at a great time when we were organizing, before I even had a job here, they were organizing a cancer neuroscience Banbury meeting. And the Banbury Center here is like a science think tank. And that initial meeting really brought in some of the top people from around the world in this emerging field. And I was able to sort of integrate myself very well into that group, and they were very welcoming. And almost all of them will be at this symposium at MD Anderson. And it's been amazing to watch the field grow over the past 5-6 years now. I wouldn't say I consider myself a New Yorker. I think I need to live more in the city. I actually I live in Jim Watson's ancillary apartment. So, so as you know, Jim Watson won the Nobel Prize for the structure of DNA. He's still on campus, so that's kind of interesting.
Well, I mean, I've lived in Boston for 20 years, and I'm not considered a Bostonian. You have to be third generation to be a Bostonian. So
Yeah, pretty much.
not feeling like you're a true New Yorker
Yeah,
is
I feel
probably
like
par for the course.
I am from DC, so I'll say I am a Washingtonian. Yeah.
Yeah. I think it's fascinating that you started in anthropology. We had just been having a conversation about how that kind of perspective can be really enlightening when you start to think about people and disease progression in the context of their environments,
Yeah,
how they
exactly.
respond to those environments, how those environments change them. And you mentioned fatigue, and I've often thought about fatigue. You know, there's the central aspect of fatigue, and then there's the peripheral aspect of
Yeah,
fatigue, and how to distinguish between, at least in this conversation, in the context of cancer,
yeah,
there can be the fatigue associated with thinking about
right.
enduring and prevailing against a
Yeah,
cancer challenge. And there can be sort of the effects that are peripheral on the muscle and other organs.
exactly.
And so, sort of sorting that out, I'm sure is going to take several lifetimes.
Yeah, yeah. It is. Fatigue is sort of one of those catch-all terms that
Yeah,
if you don't have a better word for something, it's fatigue.
right.
You know.
Well, lack of
Um,
energy reserves might be one,
yeah,
or accessible reserves.
I think the way that I think about it is that fatigue, mental fatigue, is not rescued with subsequent sleep. So it's not homeostatically regulated. So if you stay awake for a long time and then you go to sleep, you'll sleep longer the longer you've been awake. So it's reciprocal. But with fatigue, it doesn't matter how long you've been fatigued. No matter how much rest you get, you're still going to be fatigued. It's something that's separate from the actual sleep homeostat process.
Yeah, unless it's sort of a chronic disruption.
Yeah.
You mentioned homeostatic, and I was thinking about, and congratulations on that sleep paper. Very, very
Oh,
interesting.
thanks.
Um, but I was just thinking about these cycles of life, right? And I think circadian and sleep is very cyclic,
Oh, yeah.
and even down to not only just the diurnal aspect of it, but also the transcriptional networks
Yeah.
themselves.
Yeah.
Genes are cycling on and off.
Right. And it's not a small set of genes. It's like 10% of the genome, which is a huge amount. So,
Yeah, and so I just was thinking about that paper, and I'd like you to sort of describe the basic results of that paper, and then maybe we can sort of discuss what that might mean in terms of homeostasis and disruptions that can occur with treatment versus cancer versus both. Yeah.
right. Yeah. So we just had a paper published in Sleep on um, how chemotherapy influences sleep, because this is a common occurrence that can actually be pretty debilitating for people that have breast cancer, but also other cancers. So what we did basically was we took preclinical models of cancer treatment. So this would be mice. We didn't even do this in a cancer model. We wanted to look at the effects of chemotherapy on its own, because we want to be able to tease those things apart. And so we used a very commonly used chemotherapeutic called methotrexate. It's used often in breast cancer and other solid tumor malignancies. And it's also used in lower doses for things like rheumatoid arthritis and other inflammatory disorders. Um, and so we collaborated with Aaron Gibson's lab, who showed beautifully about 5 years ago now that methotrexate causes this sort of triglial dysfunction in the brain, which leads to these sort of chemo brain, which is the colloquial term for cognitive impairment caused by chemotherapy. And we wanted to see if that was associated also with disruptions in sleep, because sleep is very important for cognitive processes. And so we did that, where we gave mice different doses of methotrexate over time, and then we objectively measured sleep using electroencephalography, which will give you, if you've ever seen these squiggly waves that are brain waves, that's what it gives you. It's a um, sort of a summed level of activity of the entire cortex. So it gives you a good idea of what the whole brain is doing at any one time. And so you can distinguish sleep states based on the features of this these brainwave oscillations. And so what we did was put the mice through a normal methotrexate cycle, so an injection every week for 3 weeks, and then we stopped it and then waited for an additional 3 weeks and did a final recording to see if there were any lingering effects of the methotrexate treatment, because that really is what patients complain about, because these sleep problems can endure for years after treatment ends, and it's really unclear how that, that there's no, the drug is no longer in the system, so there must be some change,
Imprinting, yeah.
yeah, that's happening. And so essentially what we found is that the chemotherapy by itself, it it doesn't cause acute sleep problems. Like if you were to use it, it's not like that first night you're completely awake all night, but cumulatively over time it basically creates this fragmented sleep pattern that prevents animals from consolidating their sleep.
Does
And
it change like inflammation or any of these circulating,
yeah,
proteome levels?
yeah, that's a good question because methotrexate is not really known to cross the blood-brain barrier. So it's like how would giving peripheral chemotherapy influence things happening in the brain if it can't even get in the brain? And so we didn't look at it in this story, but that is certainly an important aspect. Right now, I would say in the past 10 years, people have really been looking at the cytokine hypothesis, because there is a lot of data on cancer patients at various points during disease progression or during treatment, that suggests that there's a elevation in cytokines that are known to influence sleep circuitry in the brain, like IL-1 beta, like TNF alpha, and IL-6. I think some people are starting to move away from that now because we still see these sleep problems in mice that lack a lot of those cytokines. So if you use an IL-6 knockout or an IL-6 receptor knockout mouse, they still have these chemo-induced sleep problems. If you use neutralizing antibodies, they still have this. Um, and so what people are thinking about is that I'm sure inflammation is playing a role, but I think what the brain might be detecting is a metabolic change, because mounting an immune response or creating a lot of cytokines is energetically expensive, and the brain is constantly monitoring energy states in the body, and a lot of the neurons that regulate uh, nutrient homeostasis and the feelings of hunger, those neurons directly control sleep. The most famous example is orexin. These are neurons in the lateral hypothalamus that are essential for keeping you awake. So if you don't have them, you develop narcolepsy. But they're called orexin neurons, and that sort of gives it away because if you inject it into the brain, recombinant orexin, it produces a very vigorous sort of eating behavior. And so these neurons serve two purposes: waking you up when you're hungry so that you can go find food. So it's really like the metabolic stuff is really tightly coupled to sleep.
Hmm.
And I think we just really don't know. We can't drill down to a single pathway or something quite yet. But
Well, I wonder, it's just super interesting. If you take like methotrexate in vitro and expose, like, say, circulating immune cells, do they start to produce not only IL-6 but maybe perhaps other cytokines? And then
yeah,
if they're macrophage type cells, they may actually migrate to the brain.
right. I think that's we are definitely interested in that. So there's maybe four ways that a sort of peripheral immune signal can be detected or integrated into the brain. That's either through direct cytokine signaling, so through circulation and then passive diffusion into the brain. There's also transporters for cytokines on endothelial cells in the brain, so there can be active transport that way. Um, Emerging data suggests that there is infiltration of peripheral macrophages into the brain during stressful experiences, such as a cancer diagnosis, maybe. Um, and then the last one is actually through the vagus nerve. And I think it's probably not going to be one or the other. It'll probably be a combination of all of these things. That's just my feeling.
It's just fascinating because you think about, treatments and these sort of off-target effects that treatments can have,
Yeah.
and how they can perhaps be in some ways very instructive.
Yeah.
And like in the HIV field, for example, there's pre-exposure prophylaxis where they give you
Yeah.
antivirals before you've been infected,
Right.
ostensibly to prevent infection. And
Yeah.
they're finding that those antivirals may actually have effects on aging pathways.
Oh, that's
So
interesting.
that just this weird stuff with endogenous retroviruses are being
Yeah.
suppressed,
Yeah.
and endogenous retroviruses are overexpressed with aging, and so there's sort of a little bit of a benefit, but there's also a lot of bane
Right.
to
Right.
that boom.
And it's also that, like, some, like, drugs can be very specific. If they're targeted. But you know a molecule acting in a tumor in a certain way could have a completely different function in the brain. Like a very famous example is the complement system is essential for innate immunity and sort of bridging innate and adaptive immunity. But in the brain, the complement system, the classical complement cascade is very important for synapse pruning and getting rid of synapses that you don't need. And if that goes awry because you're trying to treat something in the tumor, it's almost a surprise. Oh, we didn't even realize this was going to happen. Um, and so I think just zooming out a little bit helps put things in context.
That's really interesting. You mentioned synaptic pruning in the brain through the complement cascade, because there's likely to be synaptic pruning in somatosensory neurons with muscle, for
Yeah,
example.
yeah.
So does that not involve the complement? Is that because since it's peripheral, or
Actually, I don't know the answer to that question. I don't think anyone has really,
very interesting?
because it's pretty hard to study synaptic anything in the periphery because the density of synapse is just so much lower, like 5 orders of magnitude lower.
So Jeremy, I'm just curious about how generalizable the methotrexate results are.
Yeah.
Are other chemotherapeutics also potentially causing these sort of cyclic circadian disruptions? What are your thoughts about that?
Yes, I think. Well, personally, I during graduate school, I did a very similar study to this methotrexate study, but we used a combination of chemotherapies that is also used quite often, called doxorubicin and cyclophosphamide. And those drugs, so methotrexate's an antifolate, so that's how it influences cancer. And doxorubicin and cyclophosphamide, they act with a different mechanism of action. But the sleep phenotype, or the changes in sleep that was driven by these chemotherapy drugs, was more pronounced than the one that we saw for methotrexate. And that and those drugs actually are even, I mean, there's no real levels here, but they are pretty rough drugs to take. I mean, doxorubicin is well known for its cardiotoxicity. Um, Cyclophosphamide is an immunosuppressive drug, so it's. Uh, I think we're barely scratching the surface, really.
I think that's really
Yeah,
fascinating. You mentioned cardiotoxicity because uh, there was a recent paper showing that pulse rate variability, heart rate
yeah,
variability, was being affected uh, in the heart, and that this was a circadianly driven thing.
yeah, yeah. So heart rate variability is sort of a proxy of autonomic activity, this sympathetic and parasympathetic activity. Um, it's useful because you can measure it relatively easily in humans. In mice, we have the nice ability to actually measure neural activity and link that directly with cardiac output. something I've been interested in is some of the work coming out of Erica Sloan's lab, for instance, showing that cancer treatments, including doxorubicin, um, although they cause dieback of sensory neurons, so that's what is known as um, cancer treatment related toxicities. This peripheral neuropathy is very common, especially with cisplatin. But what her lab is showing that chemo like doxorubicin, um, while they cause this dieback of sensory neurons, which causes the neuropathy, they actually promote the outgrowth of sympathetic neurons, and those seem to actually help the cancer resist the therapy. Um, so we really need to like be careful or attentive to the specific subtypes of nerves that are in the tumor microenvironment, and how they even get there in the first place, what they're secreting, because sensory nerves secrete a bunch of neuropeptides, sympathetic nerves secrete also a bunch of neurotransmitters, neuromodulators as well. And sometimes they can be secreted at the same time, and there could be joint effects. So we're really just scratching the surface of the interesting sort of um, interplay between all the various cell types in the tumor microenvironment. But the
Well, and also the organs, right?
yeah,
And so you think about the heart rate, sort of these internal cycles along with neurological cycles. And I guess the question becomes, is there a way to holistically assess
yeah,
the effect of some of these chemotherapy interventions? And I think Eric, we chatted with Erica, wonderful, wonderful research,
yeah,
and her use of these beta 2
yeah,
blockers. And, those have an effect on hypertension. That's what they're
yeah,
prescribed
yeah,
for.
yeah,
And so, no doubt, multiple organ systems are being
right,
affected. And then they communicate, and they may even, and you could perhaps speak to this better, most certainly can, the whole body sort of synchronicity,
yeah,
perhaps,
yeah.
and how sleep disruption and circadian disruptions may not only just affect neural activity and cardiac activity, but maybe just sort of the alignment of
Yeah,
organ function.
that's a great point, and that's pretty much what we're that actually relates to a paper that we have in review right now.
Oh, excellent.
So a nice nice dovetail question. Um, and so yeah, you mentioned that this breakdown in cycles, particularly important cycles for humans like circadian cycles, um, which are not only our rest activity rhythm, but also rhythms in eating, rhythms in digestion, rhythms in the immune system, rhythms in how fast food moves through your digestive tract. That changes throughout the day because you obviously don't need to be, digesting a big meal in the morning when you're not even eating. Um, or overnight, for instance. So one of the circadian disruptions, that's what it's sort of called, that cancer patients experience is a blunting down of the circadian rhythm in glucocorticoid secretion. So cortisol is the most commonly known one in humans. Cortisol actually fluctuates extremely wildly throughout the day. In the morning, the levels can reach those seen in patients with Cushing's disease, which is, hypercortisolemia. And then in the evening, at the low end, the concentration can reach in the Addison's disease range, which is the opposite of Cushing's disease. And so this dynamic range of glucocorticoids follows a beautiful circadian rhythm, and it's pretty robust to other challenges. But what has been observed for about 30 years now is that subsets of patients, they lose this nice rhythm in glucocorticoid circadian rhythms. And if you then stratify patients that have lost that rhythm versus those that didn't, those that lose the rhythm, they fare much worse, they have lower quality of life, and they die sooner than those that maintain this robust rhythm.
This is just the rhythm in cortisol,
Yeah,
or
just the rhythm and cortisol. Yeah.
wow.
And, all of those studies have been observational studies in humans, where there's just too many covariates to figure out what's going on. Cancers at different stages, different molecular types of cancers, different populations of people, different diets, different ages. All these things are very hard to control for in humans. But in mice, we can control for all of those things very easily. So what we decided to do was to see, is the tumor doing this, or is it something else? Is it the treatment? Is it the stress? The psychological stress of having cancer, that definitely plays a role in health. So obviously the mice don't know they have cancer. They're not stressing out about that. They're all genetically the same, and they're all given the exact same type of cancer. And so when we do this with breast cancer, and we've done it a couple of times with different models, we see this blunting down of glucocorticoid secretion from the adrenal gland within 3 days of tumor onset. And it really keeps going until study endpoint, in our case, which is 4 weeks. What's really interesting, though, is that this blunting down is accompanied by the loss of rhythmicity in the brain. So the neurons that control glucocorticoid rhythms are in the hypothalamus. And what we have shown is that in tumor-bearing animals, they go from having a nice rhythm in their activity of the neurons to losing it, and it just stays kind of flat throughout the day and night. And what's happening is that ends up uh, stopping concerted output from those cells, and the glucocorticoids no longer have this nice sort of synchronous phase at the beginning, and then a nice drop off at the end. They just become sort of a flat line like this, and that is associated with worse outcomes in our models. And if we, I think the really cool thing is, and we have a preprint out, and I'll probably talk about it at MD Anderson at the symposium, um, is this: if we stimulate those neurons that control glucocorticoid rhythms at the right time of the day, when they would normally be active, when they are supposed to be active, then we can rescue the glucocorticoid rhythm. We can make it look back to normal, and importantly, the tumors grow significantly smaller in those animals, and it's associated with greater infiltration of anti-tumor immune cells. When we ablate the T cell components, we no longer see this response. So it seems like there's a real communication between the hypothalamus, the adrenal gland, and obviously a very famous effect of glucocorticoids is immunosuppression. So everything is sort of fitting together, but it really seems to all revolve around the timing of when we stimulate these neurons. Because if we stimulate them with the exact same stimulus, in this case, it was using a chemogenetic system. Um, if we do that just 10 hours earlier in the day, the tumors grow exactly the same. There's no difference. And so it really seems like we need to line things up, so to say. Like, we need to get everything in phase with each other to have the best outcomes. So, like, the clocks in the liver should be on the same schedule as the clocks in your brain, should be on the same schedule as the clocks in your kidney. But I think what's happening in cancer is they start to become out of phase with each other, and that just really wreaks havoc on just systemic health.
It's really interesting, the this blunting phenomenon in the neuroendocrine system. But I'm also just sort of thinking about what you said earlier about metabolism
Yeah,
and hunger, and it makes you think of the microbiome and gut microbiota.
yeah,
And in the context of sleep disruption, my guess is there's probably a microbiota shift,
yeah.
and the gut brain axis, of course, being operational in this case. Could you sort of speak to that, and how this might be something that is adding to the complexity?
Yeah, I've sort of purposefully tried to stay away from the gut microbiome and gut brain stuff just because it adds even another layer of complexity onto this, but it definitely plays a role. So, um, chronic sleep disruption, at the extreme end, if you sleep deprive people, obviously, it that we don't last very long without sleep, and the it was an open question for a long time as to why do animals die if you don't let them sleep, or why do humans die if they don't sleep for just 5 or 6 days. You get really crazy effects. And it turns out that sleep deprivation causes a ton of oxidative damage in the gut specifically. And if you let that go on long enough, it can really just cause almost sepsis like symptoms because the gut falls apart due to all this oxidative stress. And um, I don't know why the gut is so selectively sensitive to it. But along with that comes a complete, change in the diversity of the commensal microbes that live in the gut, which, we're learning every week secrete neuroactive molecules. They participate in processes we never even thought they could do. And so uh, I think what would be interesting is to sort of repeat some of this on the background of antibiotics, which get rid of the endogenous microbiome, um, to see if that is actually contributing. That would be the sort of easiest experiment I can think of.
Yeah, I mean, I just wanted to go back to the methotrexate results because they're so fascinating. Um, and the sleep disruption that's occurring and sort of the disruption in the circadian rhythms. Does that create a susceptibility to tumor initiation?
Yeah,
Ironically,
yeah, that's a great question. We even have a review paper called "Sleep Disruption and Cancer: Chicken or the Egg,"
ah,
because it's sort of asking the same question. It's like, okay, so if cancer alters sleep, is sleep, promoting cancer? Like, is there
or
uh,
lack thereof,
yeah,
yeah,
is there a feed forward loop
spider.
here, pretty much? And there is only one study that I'm aware of that has directly looked at this in a mouse model of Li-Fraumeni syndrome, which is, in this case, it was a breast cancer model where the mice spontaneously develop breast cancer. And in these mice, they all they did was selectively shift their timing every week by about 6 hours. So imagine you take a intercontinental, all across to Europe every week, and that's what they were doing. And then you're jet lagged every week, basically. And those mice that received that, they developed much more aggressive tumors and they grew much sooner. Like I think in their model, the normal animals develop tumors at like 35 weeks of age, but these ones would develop it at 25 weeks of age. And then they would, fare worse and their tumors would be more invasive. And they didn't look hardly any further than that. So they showed this wonderful finding, but then didn't dig down into the mechanisms. I'm not exactly sure why, but um, on top of, so that's the only mechanistic or kind of close to mechanistic study. Uh, in humans, there's a ton of epidemiological data, not for all cancers, but for definitely a large swath of cancers of circadian disruption, like shift work, being a real contributor to cancer development. And we still don't really know why. It's probably a combination of oxidative damage, this strange desynchronization of central and peripheral clocks, inflammation. Also, people that do shift work, they're eating at weird times of the day, so it throws your liver clock out of whack. Yeah, there's a lot of things that sort of feed into this that really just need to be disentangled.
You were talking about the chicken and the egg. So, which do you think it is? Is it the circadian dysregulation of the cancer cells themselves, or does that come next?
Yeah, so I think it's both actually. So that when I talk to people about circadian rhythms and cancer, there's two ways that they think of it. If they have um, a more neuro or psychology background, they think about sleep because that's the biggest rhythm that we all know of. But if you talk to a cancer biologist, they'll talk about clock genes in the tumor. And that is something a little bit different, but it's definitely related. So um, tumors actually can have a wide range of clock dysfunction. So some tumors, they tick normally early on, and then during malignant transformation, they become very fast clocks. So their transcription translation feedback loops occur very quickly, or they can lose the cyclic pattern altogether. And there are some labs that are looking at using tumor rhythmicity as a prognostic indicator.
How heterogeneous is that synchronicity? I mean, is each tumor cell on its own clock, or is there an attempt to synchronize across
Yeah, that's a great
these?
question.
That should be tractable with spatial transcriptomics, and
Definitely.
yeah, that's a very interesting possibility.
Yeah, in the brain, the central clock that controls virtually all circadian rhythms in the body, those cells are individual clocks. They do oscillate spontaneously at about 24-hour rhythms. But if you just put them all in a dish, they don't oscillate together unless they start producing this neuropeptide called VIP. And what VIP does is it just basically um, locks them all into the same phase so that they're all, you know,
Synchronized.
synchronized with a great output like this, as opposed to just like little sparkles happening everywhere. And so there must be a mechanism. Maybe I know Frank Winkler in Heidelberg is working on. He calls it tumor or cancer rhythmology, I think he's calling it. And because he found these really interesting pacemaker cells in the brain that seem to set the rhythm for the tumor. But it is very unclear when I say rhythms how we go from a rhythm in gene expression to a rhythm in calcium activity to a rhythm in cellular behavior, and then to a rhythm of the organism. There's so many spatial scales that you can work at this problem.
Yeah, I think that's maybe a good place to end the multiple levels of
Yeah.
analysis and challenges and opportunities. We look very much forward to hearing about some of the work that you have in press
Yeah.
and more about this more generally during the conversations at the conference itself.
Yeah. Great.
So thank you, Jeremy, so much. Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.